Our goal today is to explain the process of DNA synthesis and its importance. We represented DNA synthesis and the various proteins through common food items such as apples, peanuts, and marshmallows. We found it helpful to picture the essential parts of DNA synthesis as common household foods because it helps us remember and make a connection to all the specific enzymes and steps of the process. The process of DNA synthesis is important because without it, cellular division would not be able to occur properly. In addition, DNA synthesis also ensures that the correct genetic information is being passed to each daughter cell so they can function properly. Our research paper talks about a drug called gemcitabine, which is given to patients who have pancreatic cancer. The job of the gemcitabine is to block DNA synthesis in the cancer cells. Although this drug is well tolerated by the patients, on average, patients only live up to another 6 months. Only 25% of the tumors responded to the drug. The article didn't give a reason why it had such a poor success rate, but due to the fact that the author is looking for alternative ways of combating this cancer, makes us believe that DNA synthesis is not easy to block. This emphasizes the importance of DNA synthesis and how it plays a role in the growth of cancer cells.
DNA synthesis is a process that takes place within the nucleus. At the beginning of this process, the DNA double helix is uncoiled with no structure. The noodles represent the form DNA is in before synthesis.
Before we discuss the specific enzymes involved with replication, it is important to note that replication starts at many places almost simultaneously. These places are called origins of replication. These origins of replication form replication bubbles, and this is what allows for the speedy synthesis of DNA. Each replication bubble contains 2 continuous leading strands and 2 fragmented lagging strands. The fragments located in the lagging strand are called Okazaki's fragments. This important part of DNA synthesis is illustrated here.
If we were to zoom in on the DNA, we would see even more particular processes occurring. This picture's purpose is to serve as an introduction to DNA synthesis before diving into the individual steps.
DNA synthesis can't begin without the 3 structures represented here. These structures are helicase, topoisomerase II, and single-stranded binding proteins. First, helicase, represented by a banana chip, unzips the double helix at the replication fork. Second, the single-stranded binding proteins, illustrated by raisins, stabilize the single strands of DNA and keep them from coming back together. It is also important to note the broccoli acting as topoisomerase II and its function of working upstream from the helicase to relieve the stress from the DNA's unwinding.
Zoom in one more frame and we can see the role that RNA primase, represented by apple slices, plays in DNA synthesis. RNA primase attaches to the DNA strands and makes RNA primer, illustrated by peanuts. RNA primer is the starter for synthesis.
After the primer is laid down, celery, acting as DNA polymerase III, can come in and elongate the daughter strand. DNA polymerase III elongates the daughter strand by adding successive DNA nucleotides to the 3' end of the growing strand. It is important to note that this enzyme always synthesizes the DNA in a 5' to 3' direction. This explains why both a leading and lagging strand are present in the partial replication bubble.
After the full length of the daughter strands have been synthesized, blue M&Ms acting as DNA polymerase I replace the RNA primer with DNA.
After the RNA has been replaced with DNA, ligase can then join the adjacent DNA fragments, completing DNA synthesis. Orange peels in this diagram represent the ligase.
Before the conclusion of this video, it's important to note the proofreading mechanism of DNA polymerase III. DNA polymerase III is very efficient in its function. Its job is to correctly pair up nucleotides as it elongates in DNA synthesis. We've constructed daughter and triplet strand nucleotides to illustrate this process. As you can see, the big white marshmallows represent deoxyribose. The mini white marshmallows represent the phosphate groups, and the mini colored marshmallows represent the nitrogen bases. DNA polymerase III ensures that adenine and thymine match up while cytosine and guanine match up.
We wanted to end with a starting photo to serve as a wrap-up. The purpose of this will be to hopefully solidify DNA synthesis in your mind.
DNA has been served!